All right. Hello, everybody. In this video, we'll understand and talk slightly about recombinant technology and what it's all about. So this is just to help us as a quick recap. Uh, Hello, everybody. In this video, we're going to talk about recombinant technology and understand what this is all about. So basically, recombinant technology, it's all about cloning a piece of DNA slash a gene. So that's all we're trying to do. And basically what we're going to have a foreign DNA like this, which is going to be cut with a specific region that actually code for a gene. And this is going to be inserted into a plasmid or a different type of vector. And by now we know that the vector is what is going to carry this for some type of expression, hopefully. But some of the most commonly used vector are plasmid. So the foreign piece of DNA is going to be placed into a plasmid, and once it has been inserted into this plasmid, it's going to now be incorporated, or also known as transformed, in a bacterial genome. And as the bacterial genome replicates, it's going to replicate that same specific initial piece of DNA or gene that we wanted to have to begin with. But how do we go about it? So in a real life situation or in a lab, what you're going to have a piece of DNA like this. And let's say this DNA produces a gene that codes for this green product here. But the issue is that you want to clone it in such a way that it could go into a system that is going to make more of those green protein at quite a shorter rate. And what other product or species can we use than bacteria, because they replicate quite faster. As a result, it's going to be very easy for us to do that. And how do we do it as well? Because bacteria, they usually have plasmid, which is an extracircular piece of DNA that is not part of the initial DNA, and it's going to go there. So what we do, the first challenge is going to be into cutting that piece of DNA, because this DNA is not just going to be alone, it's going to be part of a system. This is where we use restriction endonucleases. And those restriction endonucleases, what they do is that they are very specific by looking at these sequence regions that are palindromic, which means that if you read them on this side, it also reads the same on the other side. And looking at the anatomy of most DNA, we are bound, or most sequences are bound to have a short region of maybe 8 to 14 base pair that could be palindromic, and restriction enzyme, they rely on those. And the nice thing about them is that both vector or plasmid and DNA would have this. So when cutting, your restriction endonuclease is going to recognize this cut and perform two type of cut usually. The first one is what is known as the sticky end, like such as this scenario here. The cut is going to be like this, and we have a sticky end. The other type of cut, it could be something like this. Once you have a sequence like this, it could also perform what we know as a blunt cut like this. But at the end of it, what we need to have is the same type of restriction enzyme ideally, and same type of cut on the DNA and on the vector as well. Then once the step is over, we can now take our plasmid here that has been cut with the same restriction endonuclease with our new piece of DNA and move to the second step, which is ligation. And with ligation, like you see in a ligation reaction, we just make sure that the DNA and the plasmid is actually one and is properly bound. Now after that, this is where we transfer it and we figure out how to insert it in this piece of bacteria. Do we cut as well, or what do we do? But before we move further into this step, what we should also look at is the anatomy of this plasmid. The plasmid, as you see here, would also have not only a region where one needs to insert the foreign piece of DNA, but a marker gene as well. So something that is going to tell us that this plasmid was actually inserted. So I'm going to clean here a little bit and redo the drawing. So some of the most common marker genes that we get are actually antibiotic resistant gene, because it allows us to perform a very nice selection process. So let's say this is my plasmid and this is a ampicillin resistant gene, as you can see here, and I have two bacteria. This is bacteria 1 and bacteria 2. In this plasmid, I've given this plasmid a gene that will produce a protein that will allow any bacteria that has the plasmid to survive the reaction or treatment of an antibiotic such as ampicillin. Let's say in this bacteria, which I'm going to call A, and this one is B. In bacteria A, the plasmid managed to make its way inside the cell, and here's the gene resistant section or our marker. And on this one, the plasmid did not make its way. It's actually stuck somewhere outside in that AP or tube type of situation. Now, let's just assume for a second here that these two bacteria are growing in a media like this, and this media is very rich in ampicillin. I'm just going to put it this way. This green thing here is ampicillin. What is going to happen is this bacteria multiply. Since it has the ampicillin resistant gene, it's going to survive, and this strain B, it's not, and it's going to die. And this is what's going to tell me that my insertion was actually successful. Now that we know that, the second challenge is now, how do we get the plasmid inside the DNA to begin with? This is where now this process comes into play. There are a lot of techniques that one can use. You could use a technique known as heat shock. You can also use calcium chloride or electroporation. But at the end of the day, all we're trying to do is to render the bacterial membrane temporarily permeable. Just for a moment permeable, so the plasmid can make its way. And once the plasmid makes its way, and we know that via the resistance of a gene, we now refer to that strain as a transform, because those cells are competent. And whatever did not take it, then we say these are untransformed. So that is the first section. And like we said, those transform after transformation, which is the process of putting the plasmid inside the DNA. Those bacterial cells are competent. But there's another process now that we need to test as well, which implies that insertion of a plasmid inside the bacteria doesn't necessarily mean that the gene was actually correctly inserted into the plasmid. So after transformation, the other process that we check for is the recombination now. So it doesn't always mean that everything is going to go smooth like this and you're going to have your expression. Sometimes you may have a plasmid inserted, but it's not inserted properly, which is fragmented, or for some odd reason there was a poor ligation, so you may not have the gene. So to test for the gene, we do another test, and this is usually referred to as the blue and white screening. So let's see and let's understand what is the blue and white screening. So the blue and white screening, it's just a rapid and quite an effective technique to identify recombinant bacteria. And when I say recombinant bacteria here, this is the bacteria that has taken the plasmid in the correct form. So any bacteria that has taken the plasmid in the correct form, or any plasmid, that has been done correctly, insertion, that is what we spoke about here when we do mention, or we use the word recombinant here. And with the blue and white screening, what we do is that it relies a lot on the activity of beta galactosidase, which is an enzyme that we find in E. coli. And what it does, it cleaves lactose into glucose and galactose. But how do we do that? So here's where it becomes quite interesting. What you would notice is that most first of all, let's start with the beta galactosidase itself. Beta galactosidase, what it needs a LAC-Z gene. So a LAC-Z gene is necessary in order for us to have a product known as B-gal or beta galactosidase. And what is the composition of a LAC-Z gene? So this LAC-Z gene is going to have coding information, an amine group and a carboxyl group, just like any gene that becomes eventually protein. But what happens now is that in order for us to test, we rely on this property. When we have beta galactosidase in the presence of IPTG and X-gal, which is a colorless molecule or colorless product, the combination of B-gal with these two, X-gal and IPTG, result in a change of color that gives this blue substance here. So in order to test, this is what we do to the plasmid and the competent host as well. We are going to have a plasmid that looks like this, and this plasmid is going to contain just the 5' sequence of the LAC-Z gene. And this is going to also have the first 146 amino acid, and that's part of the plasmid. And when it comes to our E. coli as well, because E. coli we usually find this enzyme, what we do is that the E. coli that is used is a mutant strain that is going to have this LAC-Z with a delta sign 15 deletion. So this deletion means that the LAC-Z in our E. coli is going to be incomplete. And in order for us to have a nice result, the presence of a blue color here, like this, both segments of the LAC-Z must combine together. So as you see in this picture, this is the 3' LAC-Z segment, and this is the 5' LAC-Z. One is coming from the E. coli here, and this one is coming from the plasmid. As soon as they come together, the N-terminal and the C-terminal of our LAC-Z gene, that's the only time that they're going to have the change in color, which is blue or blue colony. If those two don't come together, then we are going to have no X-gal in the solution interacting with beta galactosidase. As a result, our colonies are going to remain white. You can rewatch this again, and until you understand it, then you're going to fast forward the video and move into the second step. Here's what happens now. In order for us to make sure that this is actually what's going to happen, we are going to have our competent E. coli. By now we know what is competent, which means that they were able to take on the plasmid. Then you're going to have your media culture that is going to contain X-gal and IPTG. And on this side, this is going to be your plasmid that was taken off, and this is your foreign DNA. This foreign DNA ideally is inserted in a region where the N-terminal of our polypeptide will be disrupted. So this one should be disrupted. The N-terminal is disrupted, And once it inserted like this in this ideal scenario, it means that the LAC-Z here would not going to be useful, and as a result it will not react with XGAL and IPTG. Then we are going to get white colonies. But should our foreign DNA enter in another place, in another orientation, where the N terminal of the LAC-Z is not disrupted, what we're going to see, this molecule 1 and 2 are going to interact, and we're going to have blue colony, which is not a good thing in our case, because there's going to be no expression, and that's not what we're looking for as well. And sometimes you could also have a situation where there was ligation, but the foreign DNA did not even make its way inside the plasmid, although the cells were competent. And what we're also going to get is blue colonies, but these blue colonies are actually not what we are after, because what we want our foreign DNA in the correct orientation, and that correct orientation implies that it needs to disrupt the N-terminal polypeptide of the beta galactosidase. So that's all about the blue and white screening, and a little bit on how plasmid works.